Good afternoon, everyone. My name is Vanessa Smith, and I'm a board member for the NBIA DA. I began volunteering around 2018, 2019, and my first conference was here in South Carolina in Charleston in 2019. And so that kind of ignited my passion for genetics and led for me to pursue a career in genetic counseling. And I'm excited to present on a topic that I'm really passionate about, which is genetics. I just completed my master's in genetic counseling at Vanderbilt University, and starting in the summer, I'm going to be starting a job at Children's Hospital of Philadelphia, working on BPAN research. So for today's conversation, we're going to walk through some key concepts in genetics at a very low level and hopefully easy to understand. And we'll also explore some results reports and kind of make sense of all the terminology that's on there. And I'll share resources, including how to find a genetic counselor near you who can help personalize this information to your specific family. So here's just an overview of what I said we're going to go through, talking about DNA and genes and inheritance, followed by a conversation about how genetics may contribute to the severity of a disease, and then a brief comment on family planning. So starting with DNA and genes, I'm going to start with some of the definitions that you might already know or you might have heard, or you're thinking back to high school biology. DNA is just a long molecule that contains the instructions for building and running all things in your body. There's specific sections in DNA that are called genes, and these are what give the instructions to make what we call a protein, and that protein performs a function in your body. The collection of all of the DNA together is called genome, and that includes the genes that code for the protein as well as the non-coding regions of the DNA. This is kind of like a picture representation of the different levels of complexity. So on the left side of the screen, you can see that there's a cell, and every single cell, the trillions that we have in our body, has a complete copy of all of our DNA. And so we have the DNA level, and then it goes on to show the sections that are called genes, and that gets translated ultimately into the protein that you see at the end. So in order to kind of make this a little bit more understandable for people who don't necessarily have a scientific background, or it's been a long time since they learned this in school, I like to use an analogy called the genetic recipe book. So it's like a giant recipe book that contains the instructions our body needs to grow, develop, and function. So each gene we can think of being like an individual recipe, and we have 2 copies of each of our recipes, which we have over 20,000 of in our books. And while all humans share the same basic set of recipes, there are small variations in the ingredients or the instructions that are what make us unique, and in some cases can cause disease when there are changes or typos in the recipe that disrupt the final product or whatever you're trying to make. So if we open up the book, we can see that the letters of the book are what we call in genetics individual base pairs. There is 4 letters that make up this language of DNA, and that is A, T, G, and C. So our DNA is just a long strand of different combinations of those 4 letters, and it's very, very long. There are different ways that changes to the instructions can make a big difference in the body's ability to follow the recipe. In some cases, the recipe could be just completely missing from the book, which we call a deletion. Sometimes there's just 1 or a few letters missing. Sometimes there's large sections that are duplicated. And sometimes there's only 1 letter that is switched out with another. These and other types of changes completely change the way that recipes are read, which can cause it the product that it's meant to create to not work in the body. So like I said, this language is made up of A, T, G, and C, and all the words in this language are made up of 3 letters. So 3 letters code for an amino acid, which is the individual pieces of the protein that all together make up the protein. So like I said, groups of 3 letters make up a single word, and how we translate that, this looks very confusing, but it's actually just a way to translate 3 letters into a specific protein. So if you had a word in your DNA that said UCG, you start in the middle and you see the green U, and then the yellow C, and then you go outward and you see the G, and that translates to serine, which is a type of amino acid. And you just keep on translating each of these 3 letters all the way out, and then you'll get the sequence of the amino acids, which makes up the protein. This is an example of if I were to use that wheel to translate each of these words into what they mean in amino acids, you would get this sequence. And together that forms the protein, and this is actually the WDR45 gene, and that's the protein that it creates and what it would look like through a computer model. So a way to simplify this concept is to go back to our recipe book and look how changes to the recipe can impact the final product using our own language with 26 letters. So in this example, we have a line in the recipe book that says, add 1 egg and mix. Sometimes there are deletions, and an example of a deletion could be taking out the word 1. Now it just says add egg and mix. Well, ultimately that means the same thing. And so your body adds an egg, hypothetically, and there's no end product change. We all have differences in our DNA like this that make us unique, that don't actually change the way that the body functions, but it's just part of our genetic variation between person to person. Sometimes changes do make a difference in the final product. If you were to take out and mix, for example, that would cause you to have a bowl full of ingredients that weren't mixed at all, and then the final product would be completely different. So this isn't exactly how DNA works, because we're not like having instructions and mix, but it's just an example of how words in the sequence can make a big difference in whether or not those words are there, and if that is translated into the peptides that make up the ultimate protein. So the ingredients weren't mixed and the final product is changed. In this example, we have a deletion of 1 letter, which is the letter O from 1. Since all words are made up of 3 letters, all of them get shifted over. So then it's NEE, GGA, NDMIX, and it doesn't necessarily really make sense anymore. In genetics, we call this a frame shift mutation. So it could be 1 base pair that's deleted, 2 base pairs that are deleted. If you have 3, then you would just be missing that amino acid. It wouldn't necessarily shift everything over as far as changing what all the amino acids are after that, but the point is that just deleting a single base pair can completely change the way the rest of the recipe is read. And cause it to not make sense anymore. And depending on where that change happened, if it happened at the very beginning of the gene, that would cause changes to be made to the entire recipe, and they wouldn't be able to be read. If it happened later on in the gene, further down the line, then maybe the change wouldn't impact the protein as much as if it happened earlier. Here's an example of a recipe with a substitution. It says add 1 EFGA and mix. Well, the body's not able to read that or know what that is, so they don't add the egg, but they still mix. And if you think of cookies, if you were to not have added eggs, that could make a really big difference in how they were going to taste or look. And then a duplication is when either a small or a large part of a person's DNA is copied more than once. There can be large duplications of really long sections of DNA, or it can be small. In the example, the word one is duplicated, and that gets translated to 1-1. I had to come up with this, so I'm just trying to be creative to show it, but 1-1 would be 11, and then they would add 11 eggs, and then that would be causing a complete difference in how the end product of cookies turned out than if you just did one egg. And it may not just be one letter or one word that's missing. It could be entire sections of DNA that are missing, or sometimes even the entire gene. And that's pretty common as well. The next thing we're going to talk about is deciphering test results, because at one point or another, I'm sure you all have gotten test reports for your child or yourself, and you're like, what is all this information? It all doesn't really make sense because there's all this terminology. So we're going to try to break down some of the things that are on those test reports to hopefully help you understand your own results better. So this is an example from Ambry Genetics. I'd say that's one of the most common labs. There's also GeneDx and Invitae and lots of other labs that you could get. In the top left, you see the test that's ordered. And sometimes that might be an exome. An exome means that it's only looking at the protein coding part of the genes. It could be a genome if you've been tested more recently, and that's basically all of the DNA that it looks at. Or it could be a panel where it looks specifically at certain genes that are related to the symptoms of your child. So there's epilepsy panels, developmental delay panels, things like that. Regardless of what the test was ordered, it will say, oh, you could see that, okay. It will say positive or uncertain right here, or negative, depending on maybe you did a panel earlier on and it didn't include the gene that your child has a change in. But this will be the result, and it will list out what the gene is, or what type of result it is. So in this case, there were looking for both additional and missing pieces of DNA, as well as changes to just single letters, and also mitochondrial genome, which isn't as relevant because none of the NBIA disorders are from the mitochondrial genome, but that might be something that was ordered with your genetic testing result. And then it will have these results that tell you the specific nomenclature of the variant. And that's kind of what we're going to go into, but I also want to point out the interpretation part, because that's where it kind of gets into what the lab interprets this variant as and can give you more information about how that change impacts the protein that's being made. But going into the test results in the section of this bottom section that we were talking about, in the first box we have the gene as well as the reference sequence ID. And so the example that I'm using is WDR45 because that's what I work most with in my research, and I already had everything with the variants and everything like that. So that's what I chose as the example. And you can see it says WDR45, and then there is something that says NM and then a bunch of numbers. And that's kind of like the addition of the recipe book. So the lab uses a reference genome, or a reference of a recipe book that is a working copy in order to see where the change is in comparison to that. So this is what we researchers typically use now, but if the genetic testing was done a little bit earlier, it could be a completely different transcript. Both of them are they're good, but you can not compare different variants from different transcripts. You have to have them on the same transcript to know whether or not your child has the same variant as another child. And then the next thing will say the relevant associate syndrome. It may say BPAN, or it may say whatever your child is what you would think of as the NBIA disorder, but there could be other names. For example, I've heard NBIA type 5, WDR45 disorder, or SENDA, which is static encephalopathy of childhood with neurodegeneration in adulthood. And that is just dependent on the lab and what they find in the literature. The next part will say what the mode of inheritance is. And in this case, I messed this up. It should say X-linked. I was copying from the original example that I was going to do. But it should say X-linked for beta propeller protein-associated neurodegeneration. But it will also say whether or not the change, sometimes it will say if it's heterozygous or homozygous. Heterozygous means that on one copy of your recipe book, you have a change, and on the other, you have a different type of change. So that's basically different is hetero, homo is when the changes are the exact same. And that comes to be important when we talk about the types of inheritance of these conditions, which we'll get into. So the WDR45 gene is about 1,600 letters long from the start codon to the end codon. But in this particular result, there was someone who had a change on the 19th letter. So this is the actual coding sequence. And if you go to the 19th letter, which is that C, the C dot means that it's talking about the DNA and not the protein. So C dot 19, you go to the 19th letter and there's a C. And if you look at the second line, that's actually what this person has. Typically, most people who have a working WDR45 gene have CGA, but this individual has TGA. And if you go back to that wheel that I showed in the beginning, TGA means stop. So that tells the body to stop reading this instruction manual. There's the go, that's always at the front, ATG. And there are several different things that could mean stop, but TGA is one of them. So anything that's beyond that does not get read at all, even though it is correct or it is working. It just does not get read or translated into protein. The P dot is basically saying what is changed about the protein itself. So this is saying that there was an arginine, which is a type of amino acid, and on the 7th one, because we're looking in 3, so 1, 2, 3, 4, 5, 6, 7, it might say TER, like termination, or it might just have an asterisk that says stop. So the rest of the entire gene from the 7th amino acid on is not read. And then finally, it'll say alteration classification. And so if you're receiving a diagnosis, it's most likely that it's going to be classified as pathogenic or disease-causing. There's a whole spectrum of different variants, and you might have other variants on your report for other disorders. They won't really report out benign or likely benign because there's not a lot of evidence to support that that could be causing a disease. But they do report out variants of uncertain significance, and those are changes in genes that there's not enough evidence to know whether or not that's just part of normal human variation or if that's something that can cause disease in a person. Typically, the reason for that is that they either haven't seen it in a lot of people before, so there's no way of knowing whether or not that's the true cause of the disease, or there hasn't been any protein studies in the lab on it at all. So it's basically a lack of information to say whether or not this is causing a disease. And so they report it out, and in the future, as we gather more information, we can figure out, in most cases, over time, whether or not these variants are pathogenic or benign. The likely pathogenic is that there's probably a disease-causing variant here. We don't quite have as much information as we do for the pathogenic, which that means that there's strong evidence to be disease-causing. This is another example where, it should also say X-linked, but this is saying on the 1022nd letter in this person's DNA sequence, there is a deletion. So there's just one letter deleted. And that changes this glycine that is typically there at 342, the 342nd word, to cause a frame shift. So we have this sequence, which is the actual sequence, and they have a G right there that gets deleted, and everything else beyond that shifts over 1, and it's completely differently translated than what was originally there. And we call that a reading frame, so we read in groups of 3, and so that's why we call it a frame shift. And if it's 1 deleted or 2 deleted, we call that shifting the reading frame. So those are just 2 examples, but obviously there's a lot of other different types of genetic changes that can happen, and learning more about that might be helpful if you are interested in it, but really there's not a way to know based on someone's genetic change for most of these conditions on how severe that they will be. But that is something that more research is being done to find out about. And there are ways to figure out for certain conditions, if there's a specific variant, how likely they are to be more severe or less severe. And when we study that and we find that out, we call that the genotype-phenotype correlation. So that's when there's a relationship between a specific genetic change or a variant, and then we see that directly correlate with the symptoms it causes or the traits it causes. There's another term called variable expressivity, and this is when you're looking at 2 people with the exact same mutation or the same variant, and they can have completely different presentations. One could be a lot more severe than the other, and it can be confusing because they have the exact same change. And then the last one is called mosaicism, what the last concept we'll cover. It's when a person has 2 or more different sets of DNA in their body because of a genetic change that happened after the egg and sperm were fertilized. So to go into a little bit more details about this, I'm going to use another kind of recipe type analogy with cookies. So if we think of our genotype-phenotype correlation analogy, we have a recipe for cookies in our recipe book, but different variants of the same recipe can yield different results. Sometimes if we know the specific variant, it can be helpful in predicting. So if we were to add cocoa powder to a chocolate chip recipe, that's the variant, we can very much expect the cookies to turn out darker. And so knowing the exact change and what it causes is kind of like having a genotype-phenotype correlation. This variable expressivity, like I said, is how the same instructions from the same recipe can yield different results. So there's other outside factors that impact the way that a person grows and develops and can impact the severity of the disease that they have. For our cooking analogy, it could be like the oven temperature, the ingredient brands, the humidity in the kitchen, and personal touches. When we go to humans, we can think about how there are other genetic factors that we're not looking at when we look at just one gene and one variant. There could be protective factors in other genes that some people have and some people don't. And then there can be environmental factors or modifier genes, epigenetic factors, which are things that change based on environment, and there can even be lifestyle and socioeconomic factors. An example of this is early intervention is shown to really improve the outcomes of a lot of different things, and so severity can be impacted even if there's a genetic phenotype to a point, and not every kid with the same gene change is going to have the same outcomes. Okay, so mosaicism happens, and if your child has mosaicism, it would say so on the report if it was able to be detected in the blood. But it occurs when there is an error in DNA replication in the point of development after fertilization. So if you think about a recipe, say if the recipe called for chocolate chips, and at some point as you're creating the cookies, you mix in white chocolate chips. The result is a batch of cookies where some only have white chocolate chips, some have both, and some have just the regular chocolate chips. So mosaicism is like the same way where a person's body is made up of a mix of cells, some with a genetic change and some without. Depending on when that happened during development will likely predict what mosaicism level they have. So timing matters. If you were to have added the white chocolate chips earlier on in the process, then you would likely have more cookies with white chocolate chips. But if you added them late, then it would be less. This captures kind of how mosaicism works, but it's important to note that getting a mosaicism level or percentage is not necessarily going to predict the severity because, one, we're only looking at the mosaicism in the blood because that's the tissue that we do the genetic testing on, and that can be completely different than the mosaicism level in the brain, which is very difficult to get tissue from, and we wouldn't want to do the genetic testing at that level. So it's going to be different, but in general, people who are mosaic, because they have some copies that are working in their body, tend to be a little bit less severe or way more severe, depending on their mosaicism level in their brain, than people who have the mutation in every single gene of their body. And then the next thing we're going to cover, I know it's a lot of information, but inheritance is a key thing when it comes to genetic counseling, because a lot of people come in wondering, how did this happen, and what are the chances that this would impact future pregnancies? And so some of the key terms that we're going to talk about are different types of inheritance, like autosomal recessive, autosomal dominant, and X-linked. And then we're going to talk about de novo mutations and how those are a little bit different than these inherited conditions. So like I said, we have 2 copies of every recipe. So if you think of an open book, you can think of one side having the recipe that you got from your mom, and the other side having the recipe that you got from your dad. They could be the exact same, or they could have differences. And when a condition is autosomal recessive, that means that there is a change that is harmful on both copies that have come from mom and dad. So in order for this condition to happen, you have to have changes on both copies. And the parents are considered carriers because they have a change on one of their copies and not a change on the other. And it's actually pretty rare not to be a carrier. Everyone is carriers of different conditions. It's just part of being human. But it's rare for two parents to come together and both be carriers for the same thing, which is why these diseases are so rare. And some of the NBIA disorders that fit this description of this type of inheritance is PCAN, PLAN, MPAN, FON, and COPAN, ceruloplasminemia, Kufer-Keb, and Woodhouse-Sakakis. So a lot of them come a lot of them occur based on an autosomal recessive inheritance. And another way you might remember it from high school is using the Punnett square. That's a way a lot of people have an easier way of thinking about it. So if in this example, this Punnett square, if the lowercase c was the copy of the genetic material that had the mutation, then there's a 50% chance for each parent to pass down that lowercase c. And so 50% from mom, 50% from dad, you multiply that together and it's a 25% chance for each pregnancy to be affected, which is why there could be some unaffected siblings, but it is important to note that some unaffected siblings can be carriers themselves as well, and they can consider genetic counseling when they get to the point in life where they're deciding to have children, and they can go with their partner and see if they're a carrier or if their partner is a carrier. Autosomal dominant is less common in the NBIA disorders, but it's when there is a condition that if you only have one copy of the gene not working, it still causes disease. You need both copies to be working. You need both sets of recipes in order for the body to function the way it needs to. Typically, these are a little bit less severe because they get passed down, or they are de novo, where it's not coming from a parent, but they have a change that is new in the child, and that single change is what causes the disease. This happens sometimes in MPAN, and as well as neuroferritinopathy. The next example is X-linked, which is BPAN. So when we're looking at sex chromosomes, males typically have one X and one Y, and females typically have two X's. And so a lot of times with X-linked conditions, females are sometimes just not affected at all, or they have very minimal symptoms because they have two X's, and it kind of makes up for it. With more severe conditions, having only one X and having the disorder is not as easily survivable, or the pregnancy does not continue to full term. So with BPAN, there tends to be a lot less males, and that's thought to be because any of the spontaneous, because these are all de novo or new in the children, or they're mostly new in the children, the pregnancies don't make it to term, or the pregnancies do, and sometimes the children are very affected, or they have mosaicism, which lowers the levels of WDR45 mutations in their body. It might not always show up on the test report because you're looking at the blood and maybe in their brain they have a lower level, but that kind of explains why there is less males than you would expect for a typical X-linked condition. And then the picture on the left shows a de novo example, so parents are unaffected and the child still has that purple band, which represents the mutation in the WDR45. And then with males, or this is a male because it has one X that this child got from their father, and then one Y from the father and one X from the mother. And then this is the female example where one X is from the father, one X is from the mother, and then there was a de novo change, or a change that was new in the child that was not in the parents, that just happened spontaneously, and that child was affected as a female. There's additional layer of complexity because we have two X chromosomes in females and one X chromosome in males. Females have a mechanism to inactivate one of their X chromosomes, and so that ensures that both males and females have the same amount of X-linked activity. So this happens really early in development, and it's typically random, where in each cell it's determined that there is either the maternal or the paternal X that is silenced in the cell. And basically the proportion of the amount of cells that you have that have the mutation versus the ones that don't have the mutation can determine severity as well. And the variable presentation could be due to preferentially inactivating the cells that have the mutation, which cells sometimes do, but it's all random unless there's a big, it's hard to explain, but it's an event that leads to preferential inactivation of one of the X's. And like I said, genetic does not necessarily mean inherited from the parents. As we were talking about, the de novo mutation is when their change was not found in either parent. I'm guessing that most people, when their child had genetic testing, they got themselves tested as well to see what changes were in their own genetic information on that gene. And this is helpful information to know because it can help inform the recurrence risk or the chance that it impacts another pregnancy. So when the egg or sperm was forming or very early after the baby was conceived, there's a change and it's not actually in any of the parents' gametes or sperms or eggs, then that's when it's considered to be de novo, and the chance for it to happen again is a lot lower, which we'll go over a little bit in the family planning section, but we're already running out of time, I think. Or we have a little, yeah, we're running out of time, so I'm going to go a little bit quicker, but the main thing is that genetics can impact future family planning, and so some general information that can be helpful is to talk about how to find a genetic counselor if that's something you're interested in, and key terms that are related to family planning. So there's a website called findageneticcounselor.nsgc.org, and you can put in your location, whether or not you want telehealth or in-person appointment, and basically what state you're residing in if you're telehealth, because that also makes a difference if the genetic counselor is licensed in that state. And that will help you find somewhere that you can go, and you would bring your child's test report. That's really important so that the genetic counselor can look at the specific mutation and give you information that's accurate and can help you understand the results as well as the implications for family planning. We kind of talked about this already, but when it comes to autosomal recessive conditions, there's a 25% chance of having an affected child each pregnancy. That doesn't change each pregnancy, but there are some options that you can do either prenatally or postnatally to, have different family planning avenues. And then the other thing I mentioned before is unaffected siblings may be carriers, and so they can reach out to their own genetic counselors when they're ready to have children. And it's important to bring the siblings' genetic testing reports so they can look specifically for that variant. They could also get general carrier screening to see if they're carriers of any other type of condition. The de novo conditions, the recurrence risk is often estimated to be 1% due to the chance for a parent to have germline mosaicism. So BPAN, for example, is often thought to be de novo in most all cases, but there are some siblings that have it. And there's a high chance that the mother has germline mosaicism and that she only has the change. She's mosaic, but she only has the change in her gametes or her eggs. And there could be eggs with the change and eggs without, and in that case, she can do some IVF and embryo selection in order to select a embryo that doesn't have the mutation if that's something that they wanted to do. That's also true for autosomal recessive carrier couples. They can pursue that route, and talking to a genetic counselor is a good first step, or going to a fertility specialist. After a baby's conceived, there's ways to do genetic testing, and I know some people consider this even in cases where their child is deemed to be de novo, and it just gives them peace of mind and helps them prepare or make choices, and that is an option for pregnancies after 10 weeks. The CVS is where they take a piece of the placenta and do the genetic testing, which they could do between 10 and 13 weeks. And then the amniocentesis is around 16 weeks, and that takes some of the amniotic fluid from around the baby that has some of the cells that they've shed, and looks at the DNA and sees specifically if there's any genetic changes related to the condition that is of concern. And then there's also genetic testing at birth that can be done, and a lot of people make that choice as well. Thank you. Does anyone have, we have 45 seconds, but does anyone have questions? Thanks. I hope it was easy to understand with the analogies, but I know it's a lot of complex information too. And I think I put on the next slide some helpful links. There's this link for reading test reports. It goes over what the different sections of the test report mean. And then finding a genetic counselor near you, there's that link. And then one of the best places to get information about the genetics, it's a little bit more complex because it is tailored towards health professionals, but maybe you could copy and paste it in ChatGPT and ask it to put it into more simple terms. But that's called gene reviews. And I know for sure there's gene reviews for BPAN and PCAN for sure. And there probably is a general page for NBIA disorders that would have more information about the other disorders as well. Okay. Thank you. Oh, that was so nice. Confetti.